In this video, I will try to cover basic concepts regarding vaccines. The first concept we need to know is about incubation period. Incubation period is the time interval between entry of pathogen and first time appearance of symptoms of disease caused by that pathogen. So, the time interval between entry of pathogen and appearance of symptoms is what we call incubation period. Second concept is the difference between the vaccine, vaccination and immunization. Vaccine is nothing but biological preparation of known pathogen. A known pathogen is taken, it may be modified, it may be killed, it may be made inactive, or it may be weakened down to a state where it cannot cause disease any longer. So, this biological preparation itself is called vaccine. Administration of this vaccine to a person is known as vaccination. So, vaccination becomes intentional exposure of that known pathogen which will lead to immunization. And immunization is development of memory cells against that known pathogen. So, the whole concept is we take a pathogen, we modify it, we killed it, we weakened it, and we make a preparation which we call vaccine. We administered that preparation into a person, we call it vaccination, and vaccination leads to immunization. There will be development of immune response, there will be development of memory cells against that known pathogen. So, a person becomes immune or resistant to further upcoming infections from that same pathogen. This is the basic concept of vaccination. This concept was pioneered by Edward Jenner. He was working on cowpox virus and he was working on smallpox disease. The cowpox virus shares the epitopes with the smallpox virus, and that's how the first smallpox vaccine was made. We will talk about this concept, we will talk about this story in a separate video as well. For now, to understand vaccine, we must know another concept which is regarding innate immunity and adaptive immunity. In case of innate immunity, it is non-specific. In case of adaptive immunity, it is specific. So, what do we actually mean by this specificity? In case of innate immunity, there is no recognition. The pathogens, the antigens are not recognized, but still the immune response is being carried out. That is innate immunity. So, same response will be made against every pathogen. There will be primary defenses, the first line of defense, which is skin, which covers our whole body. The openings like mouth, nasal cavity, buccal cavity, eyes, all these cavities are protected by mucus, are protected by saliva, tears. Tears has a specific enzyme we call lysozyme, which will break the bacterial cell walls. There is temperature of our body, there is pH like stomach. Each of these factors will make the primary defenses, and innate immunity is present since birth. Whereas in case of adaptive immunity, there is specific recognition of antigens and there will be specific immune response to each antigen, to each pathogen. That is what adaptive immunity is. And to make such a specific response, we need a preparatory time. And that preparatory time is what we call lag phase. So, innate immunity was immediate, whereas adaptive immunity takes time. It may take 1 to 2 weeks to occur. That is the main difference between innate immunity as well as adaptive immunity. Another concept we need to know vaccines is the state of host immune system. If the host immune system is active, we call it active immunity, and if host immune system remains passive, remains inactive, we call it passive immunity. Active immunity is the usual case of this innate and adaptive immunity, but in case of passive immunity, the host cells or host immune cells like B cells, T cells stay inactive. Still, the antibodies may be present. We take the common example of a newborn. In case of infant's body, there are maternal antibodies which are passed down by mother through placenta. The antibodies are present, but the cells of immune system of the baby are still inactive. So, immunity is of two types: innate immunity, which is non-specific, adaptive immunity, which is specific. So, adaptive immunity is further classified as natural adaptive immunity and artificial adaptive immunity. In case of natural adaptive immunity, we have again active immunity as well as passive immunity. Active natural adaptive immunity is the normal immune response. Suppose an antigen enters your body, your body will initiate primary immune response, and it will switch over to adaptive immune response, and B cells will be active, T cells will be active, cellular response will be there, and antibodies will be made. Even memory cells will be made. This is active immunity. It is the natural course of infection and natural course of immunity. Whereas in case of passive immunity, we talked about maternal IgG in fetus. And these maternal IgG are against unknown pathogens. These are not against specific pathogen because these are derived from maternal body itself. This is passive immunity. The concept of vaccines came from artificial adaptive immunity. And artificial adaptive immunity is again of two types. It may be active immunity or it may be passive immunity. So active artificial adaptive immunity is concept of vaccines. We administer a known preparation of pathogen into body. The known preparation we call vaccine. The administration of that vaccine we call vaccination. And induction of immune response, generation of memory cells, we call that immunization. So immunization through vaccines is a form of active adaptive artificial immunity. So a known antigen is introduced before natural entry of that same antigen. So this is preventive in nature. Whereas in case of passive immunity, the immune system remains inactive, but it is artificial passive adaptive immunity. So immune system will always remain inactive, but we will supply the antibodies externally, and these antibodies are supplied against the known pathogen. For example, snake venom. Known antibodies are introduced after the natural entry of antigen within the incubation period. What does this line actually mean? We will describe in a bit. The ultimate goal of vaccination is to generate memory cells so that the person will be immune to future infections from the same pathogen. So let's take a condition. We have two pathogens. We have pathogen A with short incubation period, let's say 4 to 5 hours, and pathogen B with having a very long incubation period, let's say 10 to 12 days. What kind of immunity should be administered against pathogen A and pathogen B? We are coming to concept of the immunity against short incubation period pathogens and long incubation period pathogens. If the incubation period of pathogen is very long, we give artificial active immunity, which is vaccination. The concept behind this is that if the incubation period is long, let's say 10 to 12 days, we administer vaccines, and the immune response generated by that vaccine will be shorter than the incubation period of the pathogen itself, so that we will generate memory cells before even the immune system generates a response naturally to that pathogen. It is mostly used in case of preventive cases. We will introduce the vaccine even before the exposure of pathogen naturally. So if a person is going to some kind of forest where there is high probability of getting infected due to contaminated water or contaminated food, we give that person a specific vaccine against salmonella. So now his body will make specific memory cells, and when the body of that person will come in contact with salmonella, there are memory cells already present and they will initiate the immune response, and the pathogen will be neutralized and there will be no disease. That is the basic concept behind the vaccination, because vaccination is usually preferred with the long incubation period pathogens so that memory cells will be generated, and if the person is still infected with the pathogen naturally after that, these memory cells will kick in their action and start the immune response so that the person becomes immune to that upcoming pathogen. But what will happen if there is very short incubation period, like in snake venom? If the snake bite has occurred to a person, there is very short incubation period. Symptoms will vary according to different type of snake venom. Maybe it hemotoxic venom or neurotoxic venom, but in 5 to 6 hours there may be death in case of some deadly snake venoms. So we cannot administer the vaccine in case of snake venom. We do not have 2 days. We will artificially supply the specific antibodies regarding that snake venom, and usually we administer the heterogeneous antibodies, which is mixture of antibodies directed against different snake venoms. In that way, we are supplying artificial antibodies to neutralize the pathogen because the incubation period is very short. So this is the main concept behind the active artificial immunity or passive artificial immunity. And this is the basic concept behind the vaccination as well. So let's talk about some fundamental differences between artificial active immunity and artificial passive immunity. Artificial active immunity we also call vaccines, and artificial passive immunity we also call artificial antibodies. So, what is the natural course of pathogen? The natural pathogen may still not be entered. It is preventive kind of immunity. We give the vaccine before the natural infection has ever happened. That is the whole point. We generate the memory cells in advance so that when the pathogen will enter, there will be the immediate immune response and there will be no disease. In some cases, vaccines are still given after the entry of pathogen. We will talk about this concept in a while. But the main concept right here is the artificial active immunity is administered when there is even not the entry of pathogen. It is preventive in nature. Whereas in case of artificial antibodies, which is passive immunity, the pathogen has already entered naturally into the body and the pathogen is having very short incubation period. We cannot wait for memory cells to be formed, so we will supply these artificial antibodies externally. That is the whole point. And the incubation in case of the vaccines, the incubation period of the pathogen naturally is usually long or very long. What is the artificial administration? What are we giving to the patient? The artificial administration in case of vaccines is the known antigen. Known antigen for the disease which we are trying to prevent. And in case of the passive immunity, we are administering known antibodies against which we are saving the patient. For example, antivenom for a particular snake or heterogeneous mixture of various kinds of antivenoms we can administer in case of passive immunity. So, what will be the kind of host immune response? In case of vaccines, there will be B cell response, there will be T cell response, there will be generation of antibodies, there will be generation of memory cells. Whereas in case of artificial antibodies, there is passive immune response. There is no activation of B cells, there is no activation of T cells, there are just antibodies we supplied externally which will do all the work. Let's talk about memory. In case of vaccines, memory is developed. The memory cells, memory B cells, memory T cells will be developed. Whereas in case of passive immunity, if B cells and T cells are not active, memory will not be developed. Longevity of the immunity. The vaccines are providing long-term immunity against pathogens. Booster doses may be required, but immunity is fairly long-term. Immunity may last from years to decades. In case of the passive immunity, the immunity will last very short term because there is no memory involved. Examples, we can take salmonella, cholera, polio and all other vaccines we will talk about in this video. And in case of passive immunity, examples, we can take snake bite, we can take dog bite, hepatitis, we can take measles, we can take rabies, varicella. To administer vaccines, the person must have a functional immune system because B cells and T cells are going to be active and that's how the memory cells will be generated. The work is entirely done by the host immune system. So there is a well-functional host immune system is required in case of vaccines. Whereas in case of artificial passive immunity, it doesn't really matter if the immune system of host is active or not. The artificial passive immunity can be given to immunocompromised patients as well. But in case of vaccines, we cannot administer vaccines to immunocompromised patients. Because if we administer live attenuated vaccine to immunocompromised patient, there is a fairly high chance that the pathogenicity may return and the host immune system may not fight against that pathogen effectively. So there is a chance of disease if the patient is immunocompromised in case of vaccines, but there is no such risk of immunocompromised patients in case of the artificial passive immunity. So this is the basic concept or basic differences between artificial active immunity and artificial passive immunity. Now come to classification of vaccines. Vaccines can be classified into two main types. First is dead and inactive vaccine. Another one is live and attenuated vaccine. As the name indicates, in case of dead vaccines or inactive vaccines, there will be no replication, transcription or translation of the pathogen. And in case of live attenuated vaccine, pathogen is very much able to replicate, very much able to transcribe and translate its own proteins. And how is this achieved? The dead or inactive vaccines are made using some kind of chemical agents, most commonly formaldehyde. For live attenuated vaccines, they may be natural, they may be subjected to temperature treatment, there may be genetic engineering or lab interventions or secondary cultures. Now, what are the advantages of these? What are the advantages of dead or inactive vaccines? And what are the advantages of live and attenuated vaccines? The vaccine is completely dead. The bacteria cannot replicate, cannot transcribe, cannot translate its own proteins. So it is relatively safer as compared to live attenuated vaccines, which means they cannot cause disease. They are more safe. In case of live attenuated vaccines, they can very much replicate, transcribe and translate. That's why they provide a better immunity than these dead inactive vaccines. But there is a side effect that if the host immune system is weaker or the patient is immunocompromised, in such cases there may be regain of pathogenicity which can cause the disease. But these cases are very rare. Even regain of pathogenicity can happen in a healthy body as well, but these cases are extremely rare. These vaccines, the dead and inactive ones, there is a requirement of booster dose because the immune response is weaker as compared to live attenuated vaccines. And live attenuated vaccines provide a stronger immune response as compared to dead vaccines. So there is no such booster dose required. The most common examples is injected polio vaccine, cholera, rabies, influenza and Hep A vaccine in case of these dead inactive vaccines. And in case of live attenuated vaccines, we have MMR, which is measles, mumps, rubella. We have oral polio vaccine. We have BCG for tuberculosis. We have typhoid vaccine. We have rotavirus vaccine, which is also oral, and we have yellow fever vaccine as well. So dead vaccines are relatively more safe as compared to live attenuated vaccine, but the immune response is slightly weaker than live attenuated vaccines, and live attenuated vaccines provide a much stronger immunity than dead or inactive vaccines, but there is a chance that the pathogen may regain its pathogenicity and cause the disease, though the chances are extremely rare. That is the main difference between dead inactive vaccine and live attenuated vaccine. Examples we already have talked about in this slide. Now let's talk about polio vaccine. Polio vaccine comes in two variants. One is injected polio vaccine. Another one is oral polio vaccine. The injected one is an example of dead vaccine, whereas the oral one is an example of live attenuated vaccine. So IPV was developed by Salk in 1951 to 55 and OPV was developed by Sabin. And if it is a dead vaccine, IPV was developed using the formaldehyde treatment, and for oral polio vaccine it was developed using monkey kidney cell cultures. There are three strains of each of these polio vaccines, which are strain A, strain B and strain 3, and these whether injected or oral, these vaccines provide against each of the strain of the polio virus. Now talking about the valency of antigen, the IPV antigen is used, which is pentavalent, and in case of OPV antigen, the antigen is trivalent, and minimum 2 doses are required in case of IPV and minimum 3 doses are required for OPV. And these 3 doses ensure protection against each of these strains, strain A, strain B, strain C effectively. In case of IPV, there is majorly T cell response, And in case of OPV, there is majorly B cell response. Because it is administered by oral route, there is involvement of mucosal immunity. There will be class switching to IgA, so abundant antibodies will be IgA. There will be also systemic response as well. Whereas in case of IPV, the route of administration is through injection, so the main immunity will be of IgG. Out of IPV and OPV, which is necessary and which is not? The IPV is slightly better, but both are required. If we administer only OPV, there is a high chance that the child will become a reservoir, because the antigen antibody complex will be formed inside the GIT. It may very much be passing through the fecal matter, and that makes the child a reservoir of this immune complex and the polio virus as well. There is a concept of herd immunity. Herd immunity is when the sufficient people or sufficient population is immune to a certain pathogen, for example COVID-19 virus. If certain people or certain populations are immune, are vaccinated, there is much less chance that the people who are not vaccinated will catch the infection. This is what we call herd immunity. If the majority of population is vaccinated, or if majority of population is immune to a specific pathogen, the ones who are not vaccinated, the ones who are not immune, have very much lesser chance to get infected, and the chain of transmission is interrupted. This is the concept of herd immunity. There is a formula which is HT equals 1 minus 1 by R naught, where HT is herd immunity threshold, and HT determines how much population is required to be immune in order to break the chain of transmission. So R naught is the basic reproduction number of the pathogen. We are taking example of measles, where the value of R naught, which is the reproductive number of the pathogen, is 15. If in case of measles R naught is 15, we put the value into this formula, we get a value of 0.93. If we convert 0.93 into percentage, we got 93%. So what does this mean? This means that HT value is 93%, and 93% of population has to be immune in order to prevent the outbreak of measles, in order to break the chain of transmission of the measles outbreak. So this is the concept of herd immunity, and herd immunity can be achieved by mass vaccinations, which we saw in COVID-19 pandemic as well. Now we will come to generation of vaccines. So we will mainly talk about first generation, second generation and third generation vaccines. The first generation vaccines were whole organism vaccines. We already talked about first generation vaccines in previous slides. It can be classified into two types: dead or inactive vaccines, or live and attenuated vaccines. We talked about differences between these two. We talked about the case study of polio virus, IPV and OPV. The first generation vaccines we already covered. Now, for second generation vaccines, these vaccines are called subunit vaccines. So, specific antigens are purified from the whole pathogen. It may be proteinaceous, it may be polysaccharide, it may be mixture of these two, and the vaccine is made from that. It can be classified as recombinant protein vaccine or polysaccharide or conjugate vaccine. The example of protein vaccine is hepatitis B, and the example of polysaccharide vaccine is HIV conjugate or pneumococcal vaccine, meningococcal vaccines. And the third generation vaccines are the most advanced till now. These are the genetic vaccines. We also call them nucleic acid vaccines. We also sometimes refer them as synthetic vaccines. And these include the DNA vaccines and mRNA vaccines as well. The first DNA vaccine against COVID virus is JICOV-D, developed by India for COVID-19. And mRNA vaccines include the Pfizer Biotech, Moderna vaccines for COVID-19 as well. So third generation vaccines are genetic vaccines, which are nucleic acid vaccines. Second generation vaccines are subunit vaccines. It may be protein purification, there may be some kind of polysaccharide purification, and polysaccharide may be conjugated with some kind of carrier protein to increase the vaccine effectiveness in this case. Now come to subunit vaccine. We will talk about toxoid vaccine. So there are different toxins which are causing disease. There may be diphtheria toxin, There may be tetanus toxin. These toxins are detoxified artificially. This detoxification can be caused by treatment with some kind of chemical agent like formaldehyde. The whole point is toxin will be made ineffective. So the ineffective form, which is the inactivated form, is known as toxoid. So toxin becomes toxoid. Toxin is a natural form which was very much active, very much capable of causing the disease, whereas toxoid is artificially changed form of toxin which will no longer be able to cause disease. So usually these exotoxins are utilized in the process. So toxins are of two types. One is exotoxin, second is endotoxin. Why is only exotoxins being utilized in production of toxoid vaccine? Why not endotoxin? So the point here is the nature of most of the exotoxins is proteinaceous, and nature of most of endotoxin is some kind of polysaccharide or lipopolysaccharide. And as we know, in case of protein toxins, IgG can be made. Class switching can be happening because first antibody generated is always IgM. If we class switch into IgG, it can be class switched into IgA if it is in some kind of secretions. So class switching can happen. There will be response of both B cells and T cells. Okay, that is the advantage of using proteinaceous nature toxin, which is exotoxin. If we were to use endotoxins, the nature of polysaccharide and polysaccharide antigens will only be utilized by the B cell response. There will be no T cell or cellular response. There will be no class switching. So it is better to use exotoxins to make toxoid vaccines. Okay, this is the concept of toxoid vaccine. Next is polysaccharide vaccine. The concept is in some bacteria there is capsular polysaccharides which are enough to generate pathogenicity and cause disease. And we isolate such polysaccharides and we make vaccine out of it, or we can conjugate them with some kind of carrier protein to make even better effective vaccine. So the concept of polysaccharide vaccines is we use the polysaccharides to make vaccines, and there are certain disadvantages of using polysaccharides. If the antigen is not proteinaceous, there will be no T cell response, there will be no class switching, there will be no memory response. So in case of polysaccharide vaccines, we have no T cell response, there will be only B cell response, there will be production of only IgM antibodies, there will be no class switching, so we get no IgG antibodies, there will be no memory response as well. The examples of polysaccharide vaccines are typhoid polysaccharide vaccines, pneumococcal polysaccharide vaccines which are PPSV23, and some meningococcal vaccines as well. Now, conjugated vaccines are also a type of polysaccharide vaccines. The only difference is that we are using some kind of carrier protein along with a polysaccharide. So there is a concept that more complex is the antigen, more will be the immunogenicity, more will be the immune response. So polysaccharides are relatively simpler. We are making it complex by attaching a carrier protein covalently. And if we make it complex, there will be more immunogenicity and effectiveness of the vaccine will be more. So for carbohydrates, which are polysaccharides, capsular polysaccharides, we saw that there is only B cell response, there is only production of IgM. But in case of normal proteinaceous antigens, there is both B cell as well as T cell response. There will be class switching, so we get IgM, IgG, IgE, whatever according to the location, and we get a strong memory response as well. So we combine both of these to get glycoproteins, and glycoproteins will have T cell response, stronger B cell response, IgM, IgG class switching as well, and we have memory cells response as well. So we are using a carrier protein to make this polysaccharide vaccine even better. There was some limitation in the polysaccharide vaccine, which were no T cell response, no class switching, only IgM, and there will be temporary response. So we are covering these limitations using the conjugation of these carrier proteins, and we are getting the T cell response, we are getting class switching, we are getting a stronger memory response as well. So that is the concept of conjugate vaccines. And examples of conjugate vaccines is HIB, which is Haemophilus influenzae type B vaccine, and The carrier protein which is used is tetanus or diphtheria toxoid. Now let's come to DNA vaccines. So DNA vaccines are up till now most advanced kind of vaccines. The vector used is viruses. Introduction is to myocytes or keratinocytes, which is muscle cells or skins. So it can be intramuscular or intradermal. And advantages for DNA vaccine is both humoral response as well as T cell response will be there. There will be both B and T cell response. No booster dose is required as such. It can be given to immunocompromised patients. There are very less chances that the pathogenic DNA will be mutated, denatured or modified. But there is certain disadvantages for the DNA vaccines, which are developmental cost and research. There is host genome complications. Sometimes host genome gets mutated, or there will be some certain kind of modification which may lead to autoimmune disorders as well. There is a difference that the pathogenic DNA will not be mutated, but there are chances of wrong integration and host DNA can be mutated, which will lead to autoimmune disorders as well. So there is it is not suitable for multiple antigen approaches simultaneously. We cannot give simultaneously different kind of vaccine approaches in case of DNA vaccines, and it is not suitable for polysaccharide antigen. If the nature of natural antigen is polysaccharide, then these DNA vaccines are not enough, and they may need additional adjuvants for the proper delivery and proper immunogenicity of these vaccines as well. The two most common examples of DNA vaccines are the Japanese encephalitis vaccine and ZyCoV-D, which is for COVID-19 disease. Talking about malaria vaccine, we have malaria vaccine for Plasmodium falciparum, because out of 5 main strains, the Plasmodium falciparum is the most deadliest strain, and it causes most of the deaths in African continent. The vaccine name is Mosquirix. It is also known as RTSS, and it is a type of recombinant vaccine which is made by the Plasmodium falciparum and hepatitis B, and it is right now in trial basis in the country of Congo as well. There are two terms which are commonly used when the vaccines become somewhat ineffective. One is antigenic drift and another one is antigenic shift. For antigenic drift, there are small and gradual changes in the viral genes, and these changes are caused by point mutations during the replication of the pathogen, and it will lead to slightly generation of new strains of that virus, and it is most commonly seen in the seasonal flu. Because the virus mutates itself, there are slight variation in various strains of the influenza virus. Whereas in case of antigenic shift, there are very major changes occurring in the viral genes. Now, these changes happen by the gene mixing, which is a reassortment between two different kind of viruses, and this will create a entirely new subtype for which there will be no immunity in people. So vaccine will become entirely ineffective against this antigenic shift. Now come to the concept of adjuvants. Adjuvants are the substances which are added to vaccines to enhance their effectiveness. That are what adjuvants are. So adjuvants do not have antigenic specificity of their own, but they just boost the response of a vaccine to specific antigen. Okay, so adjuvants are some kind of molecules which will increase the immunogenicity of weak antigens when they are given alongside the antigens. They will reduce the amount of antigen or number of doses needed because they are increasing the effectiveness of the antigen. They mostly stimulate the innate immunity. They are used in many inactivated and subunit vaccines like Hep B and DTP. So what is the mechanism of action of these adjuvants? They can act by slow release of antigen. They can act by enhanced antigen presentation by macrophages and dendritic cells, or they can also increase the cytokine production from T and B cells as well. That is the mechanism of action. Here are some examples of adjuvants, and the types of adjuvants are mainly alum salts. There may be oil in water emulsions, there may be some kind of bacterial products, there may be TLR antagonists, and there may be some kind of liposomes or virosomes. and you can study this table on your own, but you can remember these types which are important for various exams. COVID-19 vaccines. If we talk about COVID-19 vaccines, the mRNA vaccines include the Moderna vaccine and Pfizer Biotech vaccines. For viral vector vaccine, non-replicating viral vector, we have Oxford AstraZeneca, which is named Covishield in India. We have Johnson and Johnson. We have Sputnik 5 or Sputnik V. For inactivated vaccine, which are dead or inactive vaccine, we have Bharat Biotech's Covaxin. We have Sinovac, which is again named as Coronavac. For protein subunit vaccine, we have Novavax vaccine. We have a specific DNA vaccine we'll talk about earlier, which is ZyCoV-D. We have live attenuated vaccine, which is Codagenix. We have mucosal or intranasal vaccine, which is again Bharat Biotech vaccines named right here. The main component or platform, the key mechanism and examples of countries in which these vaccines have been administered are represented in this table. You can study on your own. Now, last one is HPV vaccine, which is a preventive type of vaccine against various cervical cancers. It is a kind of recombinant subunit vaccine, and it contains virus-like particles, we say VLPs, which are made up from the capsid protein of HPV. It induces humoral immunity, which will lead to production of IgG against this L1 protein of the capsid of HPV virus, and it also provides long-term protection through memory B cells. The various vaccines for HPV are indicated here: Cervarix, Gardasil, Cervabac. You can see the valency, you can see the HPV types, and you can see the manufacturer in this table as well.